just before starting, let's not start. You are all welcome to propose for presentation, not only PhD students, masters, so postdocs or PIs, whatever. We need still some people for the end of April and May also. We'll probably be out in May and June. This guy will take over. But yeah, it would be nice if you could, if you don't have to not like force people, but pressure on John, for instance. Okay. Yeah, that's all. So, and today we have Luc Zorrilla, and Zorrilla in French.
You forgot one R.
Yeah, I forgot. Sorry, I forgot an R in the name. He's a PhD. He's in second year, and he's part of the Infibio group under the supervision. No. Oh.
Yeah.
Under the supervision of Marco and also Idan. I'm not sure if there's a second.
Okay, we are in the project in common, but anyways.
Okay, thank you. And yeah, the floor is yours.
Okay, I'll go straight to the thing. So I'm working with these organisms, which are microalgae. Okay, they are unicellular microalgae, and they have this flagella. I don't know if you can see, but there are some kind of antenna there that they use to swim. And we have the same flagella in our cells, most of our cells, and also plants as well, and a lot of animals too. So that's one of the main reasons for why we study them. And I work on two parts. One is on the biomechanical properties of the flagella, and the other side on how they synchronize between them to produce a symmetric beating pattern. So I will show you. But since we have a broad audience, I thought maybe to do some kind of background before. So like the width of a piece of hair is around 100 micrometers, sorry, which is basically the smallest thing that you can see by eye, more or less. And if you go below that, then you have all the microworld. So from our cells to bacteria, viruses, etc. That we cannot see. And if you look at the biological side of it, then so this is a cube of water, but of one edge that is smaller than the width of a piece of hair, and you have a lot of things going on. So different organisms that release different chemicals. If you were there for the talk of Meda, then you probably remember a few things about this. So some organisms go towards the chemicals, some go away from predators. So there's a lot of things going on, and it's not because it's small that it's not complex. But still, why should we care about microbes? So just like a few hints, the microbiologists here will know that by heart, but anyways, 80% of life history is microbial, and they are responsible for the oxygenation of the atmosphere. So we can find them to be here, basically, and they account for 80% of the phosphor, the nitrogen, and 30% of the carbon. So they're basically everywhere in the world, like in the living, and also in these three elements. And even when there is no other kind of life, they still are there. So they live in high and low temperature and very acidic, very basic places, and at very high pressure under the ocean, or also in very salty environments. So yeah, I think that's quite convincing that it can be very useful to study them. But still, what's the connection between these microorganisms and us? So first, is food. The fermentation of a lot of things is related to microorganisms, and for human health as well. So 100 years ago, we had so bacterial and viral pathogens were the main cause of human death in the world, and now it's not the case anymore. But still a good reminder that this is current issue and something from the past. COVID, and mammals and us have, including us, have gut microbiome that is essential for probably more things than I know, but at least for digestion, and it's important for your health. Then in the industry, they're also present, and in ecology, of course, they're really involved in carbon, nitrogen, sulfur cycles, and also in some symbiosis between plants and microbes, or animals and microbes, such as the coral. So yeah, I think now we can look at what is actually biophysics and how it can help us understand these microorganisms. So why studying the physics of living organisms at all? So I tried to give like an idea from afar, like perspective on what is biophysics or why it's useful, but it's so vast that I just had to give 2 examples. That's it. So this is the way that you approach a problem. I think it's interesting. Like, what are the physics problems that organisms need to solve? For example, how do you convert light into chemical energy or chemical energy into mechanical energy? Photosynthesis is an example. Also, how does microscopic biological function emerge from the microscopic constituents? So the example of how do birds coordinate to actually do some global feature as a flock. So these are one of the questions that are asked by biophysicists. But what about microbiophysicists? So if you add physics, microbiology, then superpowers. Again, there are many things that are going on. So I will just give one example on this bacteria, on bacteria in general. So here you see them. They have some kind of helicoidal tail, which is bacterial flagellum. And the question is, how do they swim? Why do they swim like this? So we'll see that by understanding the physics of the fluid dynamics at the microscale, you can understand why they cannot swim in another way, let's say. So imagine you're in a swimming pool and you push from the wall of the swimming pool, and you can still drift for like a few meters. But it's not possible in the micro world because there is no manifestation of inertia. So this has consequences on the way that microorganisms move. And so I will explain this. So this is a cylinder with like viscous fluid inside. What happens is that we put a viscous fluid because it's analogous to what happens at the microscale. And you have a pattern. Let's use the laser here. So we turn the cylinder in this way, and the pattern disappears. It mixes out. But then if you do exactly the reverse motion, you get back the pattern. So this is because the questions of physics there are symmetric with time reversal. And this is what is happening in the microscale. So the consequence is that if you have so this is called a scallop. I think in Spanish it's called vieira. If you have a micro scallop, then it has a symmetric beating pattern. So if you look at it in a video forward or backward in time, it's the same. So it will not be moving, basically. So that means that there is no microorganism that is moving with a symmetric beating pattern. And there are 2 possibilities to move them, or more actually, but these are 2 examples: to be flexible or to be corkscrew. And be corkscrew is what we saw in the video before, right? So that's an example of where physics can constrain the way that biological function is actually made. And we actually did that in the flagella sensor. I think I'm not sure it is the name of the event, but anyway. Can you confirm me then? It was called flagella sensor.
Was the research
In the CaixaBank? The research was nice, sorry. Yes. Okay. Anyways, so we did it with Medea, and you basically take a toy and you put some kind of screw here that you put on the spring. Then when you release it, it's going to turn in one direction and it's going to go in a forward motion. But if everything is symmetric here, it's kind of hard to get it straight, but it still goes forward. Yeah, so that's what I was showing before the video. Like, you actually have this flagellum is actually attached to the cell membrane, and there is a rotor here that makes it turn in this direction, and it creates this asymmetry, the beating pattern that makes it go forward. So yeah, so that's an example of where looking at physics perspective can help you understand also the biology. Now that we have this example in the microscale, then I can go back to what I'm working on. So this is a Chlamydomonas reinhartii, and there are 2 flagella that are very thin, so it's very hard to see. But basically, they are here. Trust me if you don't see them. But what are they? So it's so they have a structure that is the same in most of the cards, or very similar. There's really few variation. So you can see here that it's an attachment basically to the cell surface. That's from the cell membrane to the outside, from here to there. And if you take a cross section, then you get this circular array of tubes that are called microtubules. That are all interrelated between them, and there's also a central pair of microtubules that are connected with the others. And the red dots that you see there are basically motors. So all along these filaments, you have motors that make one pair of microtubules here to the next one slide between each other. And the constraint between them, like the links that they have between them that keeps them kind of attached, creates the curvature locally. And is this collective motion of all of these motors that creates the beating. If you didn't understand, it's okay. I will show a video later. Yeah, so they're like, this is a structure that is highly conserved in eukaryotes. And it's also, so it's an organelle of the cell that is present in a lot of eukaryotes, so plant animals, and also in a lot of organs in the human cell. A few examples are in the brain. You have like some kind of carpet of flagella that move together to make fluid in your brain. Like it's called the cerebrospinal fluid, go in one direction, and this helps for clearance of residuals in your brain, and also to protect the brain from damages. Then you have flagella in the thalamus that make the mucus go to your nose, and an example that I think is one of the most striking ones is that you have one flagellum. You have flagella in during morphogenesis, so when the organs are being made from the embryo stage, I think. And if there is a problem with this flagella, which is called in general ciliopathies, then the heart can be, instead of being here, it would be there. So it's called situs invertus. So yeah, so it's really a broad thing that I think before studying this, I didn't know that there were actually flagella in our body, and they're basically everywhere. So and they perform variety of functions. So also the spermatozoid, for example, has a flagellum. It's probably the most famous one. So it's used for motility and also for sensing, so sensing chemicals or sensing any kind of mechanical forcing. So yeah, I think that's a clear. It's clear that if you understand this, how a few things about this structure, then it can be applied in many ways in medical, also in ecology. And it raises a few questions that we don't know yet how to answer, which are: how does the flagella beating emerge as a collective self-organized motion? So the emergence from the motion of these red motors here all along the filament. And what is the role of flagella in mechanosensing in the human body, for example? And how do flagella synchronize their beating? So to answer these questions, which are quite wide, I can try to reduce a bit to this question, which is the first part of my work. What are mechanical properties of eukaryotic flagella? So one property is the bending elasticity. So if you take a spring and you extend it, then you need to exert force to get a certain displacement. Here is the same, except that you are actually doing curvature. So you need to give a certain torque to get a certain curvature. And instead of getting a spring constant, you get this EI, which is the bending elasticity. And so it's being measured over decades, but then you also have other properties that we need to measure, etc. So why is it important? Because it's been shown that if the flagella in kidney of rats, I think, are bending when there is a flow passing in a lumen, in a tube of the kidney. And this triggers a signaling cascade so that the cell below know the force of the flow, or the property of the flow. So you can easily imagine that if it was much more rigid or much more flexible, then you wouldn't get this biological function of mechanosensing of the flow. And then also, like, the elasticity and other mechanical properties can change the beating pattern. So here I have a video to explain. Okay. So this is spermatozoid, I guess. And if you look closer at the flagellum, then you have what I showed you before. Maybe it's a bit more visual. So you have all the microtubule doublets that are interconnected between them, and these are the motors in between here. And okay, so these are the motors, but seen when the filament is like this, and they make one slide, one tube slide, tubule slide with respect to the other one, and all of this together. Let's see if I can get it. All of this together and the way they are coordinated, which is not clear yet, makes the bending, the collective motion, which helps this flagella beat, and then ultimately go forward in one direction. So yeah. So understanding the bending elasticity, other properties, can help in this bigger question of how does this collective motion, this beating, emerge. So if we reduce the question into a research question, then we get what is the length dependence, dependent viscoelastic response of the flagella of temporary force. So behind this barbaric question, you have basically this experimental setup, which is you put a cell on a pipette. We paralyze the flagella, and it's in a flow chamber, and then you make the chamber move with respect to the cell in a periodic fashion. And then we look at the flow field around the flagella. Okay, and at the same time, we visualize the shape of the flagella. So we know the response, the shape dynamics response with respect to the flow, to the flow symmetry. Okay, so I'm not working on the experiments yet because many reasons, but basically on the modeling side, I reproduced a numerical model to try to see how the shape to look at the shape as a response to the periodic flow by changing the internal parameters of the filament of the flagellum. And here we did some benchmarking. So basically, when you put a constant vertical flow with just bending elasticity in the flagellum, then you reproduce the analytical solution, mathematical analytical solution. So, and we can use this model to try to infer the parameters, okay, that give the shape. So we have to invert this equation. But when you have a lot of parameters, it's not easy, and it's also not a linear model. So it's kind of a still open question. But if you put only bending elasticity, then you have just one parameter, right? So it should be a bit easier. What you can check is that if you put a very low amplitude periodic flow, we get a one-dimensional response. So, well, here it's like PCA, principal component analysis, and you look at the spectrum of the covariance matrix, and there is only one-dimensional response. And here is the same. We do a spatial Fourier transform over time, and there is only one mode that responds to the external flow. There's a periodic response, but it's unidimensional. Well, that's what we expected. But then the idea behind this is that maybe to infer the parameters from the shape, we can look at this kind of parameters or quantities and see the dimensionality, if the dimensionality of the response changes with respect to the parameters here, and see if we can already categorize the parameters as a first idea. Because if you have only one parameter, it's not so hard, but if you have a few, then you have a multi-dimensional space, and it becomes really complicated to invert this problem. So for that to say that it's still no work. So that, yeah. Then the second part of my work is on how flagella synchronize. Why do we care about this? Because in microorganism, you have these so-called metaconal waves. So it's like a flagella carpet where they make these wave patterns. For example, to imagine like a ball of So it's to move, it's to create a feeding current generation. So it actually makes feeding current in this direction, for example, if you see the wave traveling in this way. And then in the organism that I showed you before, Chlamydomonas, then you have two flagella that beats together in a breaststroke movement, but we don't know how they synchronize. Like, it's important to understand it, but we know that they actually can couple only through the fluid. So like with, if they beat like this, then the interaction through the fluid makes them synchronize. But maybe they can also synchronize through the internal coupling, like the coupling through the inside of the cell. So that's the question here. So the setup is, so this is experimental data from last summer from a bachelor student in Warwick that I'm actually analyzing now. And this is a pipette. You put the cell on the pipette, and in front of it you have a cantilever. So like imagine you're in front of a wall on its longer direction. If you're far from the wall and you beat your two flagella, then if there is a fluid in between, then maybe there is an interaction through the fluid between these two flagella. But if you're close to the wall, like here, then you can beat as much as you want. Normally, there is no interaction through the fluid. So if they still synchronize, which in this video they do, then it must be through some kind of internal coupling. If you look at if we analyze a bit the flow field, the velocity on uniflagellated mutants. So here you have only one flagellum, and we see clearly where the maximum intensity is. And then we look at the power density spectrum. Then there is a steady beating at a certain frequency, like around 30 Hertz. And then at some point, we induce a light stimulus that makes the cell stop beating, basically, or like reacts to it. And you can really see the difference. Okay, so keeping on analyzing the uniflagellated mutant, when the flow is close to the flagellum, you see like a strong peak, and otherwise you see noise. So by doing some signal to noise ratio on each square of this image, then we see the propagation of the flow field, and see clearly that it passes through when the cantilever is far, and that it doesn't pass when it's close. So it's basically our control experiment. Like, we want to be sure that this happens. Otherwise, we say that the coupling is through the cell, but maybe it's not true. So here we're sure that if there is synchronization with two flagella here and here, but the flow doesn't pass, then we're fine. It has to be from the inside. Okay. So we can now look at the wild type organism. So we don't see much, but this is the pipette. Here you have the cantilever, and here you have the two flagella. And we look at the region on each, and you have top, bottom for the near, and then top, bottom for the far. So when you see here, there is like a synchronization of the two flagella. But at some point, we put the light stimulus. Okay, so they stop to synchronize, and then they will start to resynchronize afterwards. And but it seems that it's longer. Well, actually, yeah, it seems that it takes longer for the one when the cell is near the cantilever to restart to beat at a certain frequency. So I still have to look at this and see if I can get like a better quantification of what's happening. But that's the idea. So the conclusion of this part is like, yes, they seem to synchronize even though there is no hydrodynamic coupling. So they must do it internally. But it seems that maybe the fluid around this, the hydrodynamic coupling plays a role into making them more synchronized, basically, like resynchronized faster. Yeah, so that's more. So that's really to summarize my work until now. So you have one side that is to look at the mechanical properties of the flagella, the elasticity, for example, the bending elasticity, and the other parts to see if, how do these flagella synchronize on this type of organism. That's it. Thank you.
A very preliminary question. I think that on your second slide, you showed the reappearance of a pattern that you had put in the fluid.
Second
Right.
slide. Eh? This one? No,
No,
the second of the work. I don't understand what is second for you.
no. Well, I don't remember if it was the second. It was at the beginning.
Okay, can you
That
repeat the
steer
question?
one way and the pattern disappears, and then
Yes,
you steer
I
the other way
understand
and
now.
it reappears.
Okay, where was that? Well, actually, it's on this slide.
My question is
Yeah,
simply, does it not contradict the second law of thermodynamics?
yeah, it's a good question. Actually, there is still diffusion, but I think that maybe I didn't really. Yes, for sure, there is still thermodynamics, but I think maybe at that scale, diffusion is kind of slow. I don't know. So diffusion is a manifestation of the thermodynamics, and if you have diffusion, you wait long enough, and then the pattern, well, the R that you see, will disappear. Okay.
But at the end, it seems to me very analogous to saying that you put a grain of sugar in a coffee, you stir one way, and the sugar melts, and then you stir the other way, and you reconstruct the crystal.
Yeah.
And
But there is no
thermodynamics does not forbid it to happen. It simply says that the probability is 10 to the minus
Yeah.
65.
No, sure.
So
But here what's happening is it's so viscous that actually the layers of fluid are not mixing between each other. So they stay parallel. It's just that since you turn, you have one layer that does like this, then it comes.
yeah, but
There's
the R
very
is
small
made of
variations.
some substance.
What? Sorry.
The letters are made of some substance different. And you are saying that the molecules of such a substance reorder in exactly the same way as they were.
No, I think here is a matter of scale. Probably that if you wait long enough, it will diffuse out. So if you wait long enough, you will see thermodynamics appear again, right? Or the effect appear.
Okay,
I'm
so you
sure
are
that
not
locally-
saying that thermodynamics
No.
is wrong.
No, there is an effect. But you have to look closer, I think.
Okay.
Go with the scale. All right. Maybe one day.
Thank you for your presentation. I understand more what you are doing now. I have a question about the evolution of these structures. Do you know if, for example, a very, let's say, primitive bacteria or archaea, can you see a difference in the structure between our spermatozoid, for example, and a bacteria?
Okay, so that's something I should have said. So this is a flagellum of the bacteria. You see, it's very different from the structure I showed you in the video.
Yes.
So the flagella from bacteria are really not the same at all as the one in eukaryotes. So that answers your question.
Yes.
Then there are a lot of variations
Okay.
within this flagella itself in eukaryotes.
Okay.
Sometimes, so here you have like a circular array of 9 tubes.
Yes.
Sometimes you don't have the central pair, sometimes you don't have the motors. Sometimes, so it can collapse into a point that is smaller and you have just 2 tips that go out. There's a lot of stuff going on. I mean, I'm not saying it's always exactly the same, but the beating ones, they seem to have some structure where they keep the same rigidity because of the central tubules and the interconnection between them. Well, I would say that.
Do you think it would be interesting to do the same test benchmark on different species of
Definitely.
bacteria or cells, even different kingdom? Yeah, you will see different. Sometimes maybe it's not
Yeah.
synchronized.
Yeah, actually, we are gonna do the experiments with different mutants already. So some of them lack the motors, some of them lack another link, some of them are not interconnected between the 2 flagella. So there is a lot of stuff to try already on the same species, actually, because it's a really known species. So we also know the proteins and the mutants. So, but it's a good idea for sure. Yes.
And you said they can have like those two different tasks, so either the movement or the signal also. Is the structure, like as you said, the cross section the same for both tasks, or do they vary, like the structure itself of the flagellum depending on the?
Yeah, that's a good question. So it's related to what Samuel just said. So it's a bit of an old nomenclature. So I think there is also, it's not binary, but basically you have the active one and you have the passive one. The active one has the motors there, and you have the central pair. The passive one doesn't have the motors, and I think doesn't have the central pair. Then you also have things in between them. And the passive ones are the ones that are involved in sensing, well, in the kidney, for example. And the active ones, for example, is the one in the spermatozoid or that they use to beating in the microbiology study.
They do some mechanical sensing as well, and and other types of sensing.
Well, okay, I can say that too. Like there is also mechanical sensing in the active ones, and probably it's always a mix of both because there is also an activity in the passive ones, because maybe there is some movement from the inside of the cell that makes it's not just a random fluctuation. Like there is also some activity in the passive one. It's really not a binary.
I mean, mostly it's not like this, like, I think mostly in biology, like, of course, you can distinguish between one or the other types, but then, like, in the practical sense, I think it's always combined with active and passive. So it's understandable that there's probably a mixture on the type, but I was just wondering, so where that could be.
Yeah, so it's mainly on the motors, I would say, and also the central pair.
And sorry, just thinking about that again, it's like the number of the motors and the like the type of flagellum, are they always like the same number, or are they sometimes like just 4 or
The number of motors?
yeah, not numbers, but the one with this one.
Yeah, sometimes you can have I don't remember. I'm sure you have different names. This is the most common structure and the most studied one.
These highly conserved structures we find in 9+2 almost students.
I see.
We have weird exceptions.
On your last experiments, you clearly showed that you both of your flagellas came back to their symmetrical frequency beats.
Yeah.
Much faster when you have the influence of the hydrodynamics. And so, your last
That one.
Like, and you said it's not when you put the light shock.
Yeah.
It's not symmetrical anymore. But
So I would
cannot
like
see,
that
I'm
one.
not sure I can see well, but it still looks between top and bottom very symmetrical. The frequency bits of your coordinates description.
Oh,
it's just
actually, yeah, it's very similar actually. But maybe it's the response to the light.
they have a similar
it's-
response.
Yeah, so I mean, the next step of this
I was
experiment
about to ask, what's the next step?
is to look at the signal and to try to see if they're in phase or not. Because here I'm not actually looking if they're synchronized or not. I'm just looking at the response to the light stimulus. And I already see that, so this one restarts to beat at more or less the same frequency at the same time as this one. So it seems that, okay, but maybe it beats alone. We don't know if they're actually beating in phase or not. So what I want to check is the phase, if they're actually beating in phase. And that would actually answer your question, I think.
Yeah, because you have the amplitude, you have the frequency of the beats and then the phase.
Yeah.
But you have already what you need to do this one.
Yeah.
Okay.
Yes, Sarah?
What are the axes of?
Yeah, good question. This is the
I
time.
don't see anything from here.
Okay. This is the time on the x-axis, and on the y-axis is actually the frequency. So it's like every column that you do is like a Fourier transform.
It's a spectrum.
It's a power, yes.
And the
It's
x
over.
is time?
The x is time.
How long?
It's okay. It's 3 seconds,
Okay,
I think. Yes. And at 1 second we put this.
okay, thank you.
Welcome. You can also come closer. But I didn't put the time, so it's not. Yes.
I was just, like wondering about like a more general thing about the method, like how you, when you showed the, like the pipette and the cell, how hard is it actually to do it practically in the?
That's a very good question that I cannot answer.
Okay.
We can continue it like.
Okay, yeah. Yeah.
Maybe for future data, it will not provide.
So it breaks apart or like what is it?
No, it's not hard. The cell wall is fast, so you can actually set a little bit of the pressure and there. Takes a little bit of practice to probably generate the like that in the way that useful. They break thin, the 2 microns wide and rounded, so under the cell, and then find a way to reach them. They're clean around. So they
Yeah.
and that's one to be that.
In my mind, sorry, a cantilever is like a bar, but here I imagine that it's more like a plate.
It's a wall, yeah.
Yeah.
It's these ones we bought some standard cantilevers for AFM, tipless. So they are, what is that? I think it was 400 nanometers by 30 microns by 200 or 300 microns, something like that.
And
So the interesting thing that,
And so right now you cannot kind of prove that there is at least for sure an internal coupling for the synchronizing. So, but how to find out which exact mechanism inside the cell might do the synchronization?
That's a good question. I mean, we have some visualization of what is the structure. Okay, so it kind of continues, like you have the flagellum and kind of it's this one. You have the flagellum and it continues inside with a direction like this with an angle, and the same for the other one. There is some coupling. I don't know like what is exactly the elastic constant. I'm not sure if it's known. I don't know if it's always a simple model and check. But I think that's already if you get the phase of each flagellum and you can put a simple one-dimensional model of synchronization, like 2 pendulums synchronizing, and get back the strength of the internal coupling. Okay, that would be, I think, well, from this data, it's the most sensible thing to do, I guess. So yeah, that's good.
Is there a technique to visualize what happens inside the cell? In this case, where the cantilever is closed, because that might reveal the mechanism for coupling without hydrodynamics.
Could be. I'm not really the expert in this. I'm sure. Well, during the movement, maybe it's fun. Yes.
no, I just wanted to comment on that. The flagella stem from basal bodies. So these are structures that are similar to the cross section of the axoneme, which is this 9+2 thing. Slightly different because instead of being doublets are triplets, and these are anchored into the or rather actually generate rootlets, microtubular rootlets that keep the organelles of the cell in place. So the 2 basal bodies actually are connected with each other, between each other, by a set of fibers. So it's called striated fibers. And we know that if you cut the fibers, because there are mutants that don't have the fibers, then there is no synchronization in this case. So that shows that for a single cell, the internal mechanical coupling is necessary, but you don't know whether it is sufficient. At the same time, in the past, what we have done is to take 2 cells, not of this, but different species for a specific reason now I don't want to enter into, and showed that if you keep them in 2 different pipettes, a different separation, so they're only coupled hydrodynamically, they synchronize when the separation becomes smaller than something. And this is through a sudden node bifurcation. So now we have the evidence that the hydrodynamic is sufficient on one side, but the fact that on the other side that is not sufficient for a single cell. So what we wanted to do here was, for the point of view of the single cell, where we know that hydrodynamic is not sufficient or maybe is necessary to, in a sense, close the circle and show whether and test whether it is actually also necessary or not. So what this shows is that it is not necessary, but it might still, so this is the point that it was making before, it might still be contributing something measurable. Okay, so that's the other bits that he was talking about, and maybe it has an effect on the.
Well, I was thinking of something like, can you visualize somehow, for instance, with a confocal microscope, the bottom of the
No.
bodies and the
With a
example?
confocal, you cannot, because the in-plane resolution of a confocal is not enough to do that, right? Because it's still limited by wavelength of light. What you're doing is you are, with the confocal, you're increasing the axial resolution very much. But in this case, the problem is that the fibers are of the order of 50 nanometers, something like that. So you cannot do that. What you can,
I don't know if a confocal or
right?
also an electronic, no, because you
I know
have to
exactly,
kill
because you need to kill the cell.
cell.
So what we have been trying in the past was to try to visualize calcium in the cell. For several reasons, calcium is super important as a cell secondary messenger. And but this is a pain in the ass. Sorry. But,
Sorry, I was asking Ilan, what about using one of those 2-photon microscopes that increases about the resolution because
of course, you need to have something that fluoresces. Yeah, that was saying that if you want to insert a fluorescent dyes inside the cell, is a pain in the ass, because the cell has a cell wall,
yeah,
and it does not uptake dyes as mammalian cells do, for example. So the result is that what you can do it is through a technique it's called biolistics. Essentially, you coat nanoparticles of gold with your dye, and then you shoot them at the cells. I've talked with the person that does this in the world, but the yield is like 5%. And the majority of the cells have too much dye size, you can't see anything, and just not the correct technique. So the correct thing would be to do a genetic modification so that the cell expresses in situ in the structure that you want a fluorescent dye, the correct fluorescent dye. The problem is that CLAMI genetics is problematic because it doesn't do homologous recombination, and therefore you chop the gene, then you put in the other gene, and it just goes anywhere in the genome. Okay, so now I don't do CLAMI genetics. I know now they've started to develop some techniques, are mostly aimed at the chloroplast. Chloroplast has its own gene pool, so maybe that's the reason why they managed to do it. But in general, it's aimed. So the majority of the mutants actually are done through UV mutagenesis. That are old, so they just blast the UV and they look for interesting phenotypes and hope for the best. Sorry, a bit rude of
No more questions? No? Okay, thanks, Luke.